Biotechnology and human health. The potential to cure human illnesses and improve general health with biotechnology is immense, and many applications of biotechnology are already in use. Sequencing the genomes of tumors to identify the mutations and treating patients with targeted treatments based on the findings. Using cancer patients' own T cells to fight cancer by stimulating them to increase their number in the lab and then returning them to the patient's blood. Harnessing genetically modified bacteria to clean up toxins and other pollutants in the environment. Developing safe, highly effective vaccines to prevent infections by pathogenic microbes. And treating human diseases with proteins made by microorganisms. Diseases that are caused by one mutant gene or one missing protein are excellent candidates for biotechnology cures. If scientists can repair that gene or replace its protein in the right place and time, the disease is treatable. Multi-factor diseases are harder to understand and treat. Diabetes is caused by the low level of just one protein in the bloodstream that prevents the proper production of insulin. By using biotechnology to increase the level of this protein in the blood, diabetes can be successfully managed. In order to develop this treatment, scientists needed to use knowledge about genetics, molecular biology, microorganisms, and medicine. Here's a definition of diabetes from the Centers for Disease Control and Prevention. If you have type 1 diabetes, your pancreas doesn't make insulin or makes very little insulin. Insulin is a hormone that helps blood sugar enter the cells in your body where it can be used for energy. Without insulin, blood sugar can't get into cells and builds up in the bloodstream, which means the cells are starved of the energy they need. High blood sugar is also damaging to the body and causes many of the symptoms and complications of diabetes. And here's some further information, that website there from the Centers for Disease Control. Type 1 diabetes is usually diagnosed at a young age. Its cause is primarily genetic, unlike type 2 diabetes, which is a disease of insulin resistance and commonly develops in obese people over time. Type 2 diabetics make some insulin, but not enough to keep their blood sugar levels at a healthy level. Human insulin can be produced in a lab by bacteria, that's pretty amazing, that have been genetically modified. A simplified version of the human insulin gene is introduced into E. coli. The protein is recovered from bacterial cultures and is then purified prior to injection. The E. coli has been genetically modified to produce human insulin. Standard molecular biology techniques are used to make recombinant DNA molecules and have the bacteria express the proteins they encode. The DNA Learning Center has created a new slideshow here explaining how human insulin is made using bacteria. We'll summarize that in the next few slides. Knowledge of the desired protein is necessary to address the problem. Insulin's amino acid sequence was determined. Insulin is a small peptide hormone of two peptides. Here's one, here's the other, the A and B chain, and they are linked together by two disulfide bridges as shown. Disulfide means two sulfurs, just like that. For each peptide, A and B, a DNA sequence was synthesized that could encode it. Programmable DNA synthesizing machines are used in many labs. DNA polymerase was used to make the second complementary strand of each DNA molecule and to make many double-stranded copies of it. The double-stranded DNA encoding the peptide was then inserted into plasmid vectors. The bacterial plasmids were engineered to express inserted pieces of DNA. In addition to a site for the insertion of genes, the recombinant plasmids also carry, and hopefully you'll remember this picture from about, 30 slides back, and I said we'd talk about it later on, especially this piece. Well, here we are. We have an antibiotic resistance gene, so that any bacteria that has a plasmid vector will be able to grow in the presence of antibiotics, and you'll see why that's necessary shortly. An origin of replication, so that the plasmids will be replicated in bacterial cells, and then a promoter, so that RNA polymerase will bond to the plasmid and transcribe the inserted gene, which is right over here. The recombinant plasmids were transfected into bacteria. Transfection is a method used to transform bacteria in the lab. It enhances the movement of DNA into bacterial cells. To transfect bacterial cells, they are briefly shocked chemically, thermally, or electrically, which makes them leaky and more likely to allow DNA to enter. Their membranes recover minutes after the shock. Bacterial cells that pick up new DNA have been transformed. Transfection is carried out in a test tube with billions of purified plasmids and shocked bacterial cells. We have the transfection process, and it results in the transformed bacterial cell, and here's the plasmid right over here that came in through the leaky cell after it was shocked. And here's our gene. This is the one we want to insert into the new genome. Transfection leads to a small percentage of bacteria being transformed by plasmids. Good thing we had billions and billions of them. The bacteria are then placed on petri dishes in selection media, a nutrient-containing agar with added antibiotics. And here come the antibiotic role. Plasmids carry the antibiotic resistance gene, so only bacteria that have been transformed with plasmids could survive and form colonies in the selection media. So only the bacterial cells that carry the plasmid that we want will grow on the selection media, since the plasmid carries antibiotic resistance. So here we have our transformed bacterial cells. They're spread on the nutrient agar with antibiotic, which will kill all the cells except these. Then those cells will form little colonies of the transformed bacteria, these little circles here. Another way of showing it, we have transfection leading to a small percentage of viable transformed bacterial cells. So we have our antibiotic-sensitive bacteria. We put in the DNA plasmids. These are the cells that we want to keep. That's the transfection part. And they're over here. We've got the plasmids in there with the original DNA. We spread the whole thing on a nutrient agar with antibiotic. So only bacteria that carry the plasmid will grow on the petri dish because the agar has an added antibiotic that kills bacteria lacking the plasmid. So these colonies are the ones we want. These bacterial colonies that carry the gene for insulin A chain or B chain are expanded by growing them in large liquid cultures. The bacterial cells were induced to express the insulin genes, and they release the insulin protein into the liquid culture media. The final step in the process. We have the bacteria producing insulin A chain. Here's the insulin B chain. They're purified in this step here, and then we combine them. Okay, here's A and B. Here's our disulfide bonds that form, the little red lines there. We now have insulin that is ready for injection into human beings. Because insulin is a small dipeptide, it spontaneously folds into a shape that is recognized by human insulin receptors. And as said on the previous slide, it is ready for injection. So here's a diagram of insulin proteins right here. Now, why did this work? Well, insulin is a small peptide hormone, making it an excellent candidate for production by bacteria. Larger proteins usually do not fold correctly or get modified correctly in bacteria because bacteria lack endoplasmic reticulum, Golgi apparatus, and the proteins that help with folding. Recent advances in biotechnology allows us to produce more therapeutic proteins in cultivated mammalian cells so that they are folded and modified correctly. Mammalian cells grow much more slowly than bacterial cells, and they reach lower cell densities, so using mammalian cells is more difficult, expensive, and time-consuming. Medicine is being revolutionized by more and more targeted, genetically engineered therapies for diseases. In addition to diabetes, many types of cancer, autoimmune diseases, and some types of heart disease can be treated with genetically engineered proteins. The discovery of plasmids and enzymes that modify DNA in the 1970s began a 50-year revolution in molecular biology, agriculture, and medicine that continues to this day.